We are moving to our last speakers, János Pigler and Tamás Riesz, fourth-year medical students, winner of the second session of physiology, pathophysiology, and pharmacology. And the title of the talk will be Cytoprotection by the nitrogen monoxide donor SNAP and BMP against hypoxia reoxygenation injury in rat engineered heart tissue. Please.
Okay, thank you very much. My colleague is János Pigler, my name is Tamás Riesz. We feel a privilege to be here. We would like to present a project in that we could take part at the Department of Biochemistry, and we are very thankful for the cardiovascular research group for that, because we were involved into a relatively new research field, and we see a great potential in it. In this slide, you can see an engineered heart tissue that we'll characterize in the next few minutes. We would like to focus on three main points to introduce our work comprehensively. First, why was this new method developed? How can we utilize it? And what did we personally use this system for? Why? It has many connecting reasons. The main motivation for the cardiovascular innovations is the fact that the leading cause of death are still the diseases of the heart and circulatory system in the developed countries of the world. In addition, there's a long way from the first step of drug improvement to the clinical application. The experiences are diverse, expensive, and time-consuming. The in vitro models that are usually the first step in this procedure provides important but not enough data, especially functional ones, so they have to be repeated in ex vivo and in vivo models. I would like to show you the most important experimental systems that are used in the laboratories nowadays. The in vitro model offers the most realistic simulation. Several parameters can be monitored. However, the procedure requires professional knowledge, considerable financial background, and induces high ethical responses. The ex vivo model is suitable for measuring functional parameters. However, the main disadvantage is that it cannot be used as a chronic model. Two in vitro models are used in the cell culture laboratory of our department. We expose neonatal and adult red cardiomyocytes to simulate ischemia reoxygenation, and we test cardioprotective materials on them. Besides all of these, the engineered heart tissue can be a versatile tool for further investigations. So how could we utilize this system? Engineered heart tissues can be maintained for a long time, so they are proper for testing acute and chronic effects of drugs. Several diseases can be monitored or simulated, just like the hypertrophy model that is visible on this slide. The other approach for the application is the implantation therapy that also supplied promising achievements. And now let's see what did we personally use the system for. The generation of the EHTs and the experimental procedure was performed at our collaboration partner in Hamburg in the following way. The basis of the structure, called master mix, made of extracellular matrix components and neonatal red cardiomyocytes. This suspension was pipetted into rectangular casting molds in which two silicon posts were positioned above. The cells attached to these silicon posts and they formed an integral tissue after their differentiation. About 2 hours later, the fibrinogen in the mix polymerizes to fibrin, so the EHTs can be transferred into a 24-well plate, and a special EHT medium was added in order to enhance the development. These EHTs started to contract 8-10 days later at 37 Celsius degree, 7% oxygen, 7% carbon dioxide, and 40% oxygen concentration. These contractions were recorded with an automatic video system in a special chamber. In this slide, you can see the maturation of the EHTs that demonstrates how long they can be maintained. After a while, their histological structure is more and more similar to the original heart muscle tissue. We utilize 15 to 22-day-old EHTs. They contract rhythmically, and their structure is much more organized comparing to the earlier stages. The main profile of our department is testing cardioprotective materials for the reduction of ischemia reperfusion injury. I would like to mention 2 out of several articles that our experiments based on. A simulated ischemia reoxygenation system was set on neonatal red cardiomyocytes, and the protective effect of S-nitroso and acetylpenicillamine on mouse embryonic stem cell derived cardiomyocytes against simulated ischemia reperfusion injury was published in 2014. According to these findings, our aim was to set up and validate a simulated ischemia reperfusion model on the EHT system. We used 3 main groups. All of them received fresh medium 24 hours before the experiments. The EHTs were maintained and their contractility was monitored for additional 2 days after the reoxygenation in fresh medium. In the first group, the cardioprotective materials were added under 180 minutes hypoxia and 120 minutes reoxygenation. The second group was a positive control. Fresh medium was added besides the safe procedure. In the third line, you can see the time-matched control. No cardioprotective materials were added, and they were not exposed to hypoxia either. After reoxygenation, the media were collected and the levels of the biomarkers were measured. After the follow-up period, the media and the EHT's histological samples were frozen for further investigations. Now let's see our results. First, we would like to present visually how does an EHT look like under physiological condition. Its basic rhythm is uniform. The frequency is about 160 beats per minute. Under the hypoxia, most of the EHT stopped. The alteration of the gas concentration was enough because the EHTs are much more sensitive than the 2-dimensional cell cultures. Using hypoxic solution, we wouldn't see any recovery, as we can see in this video demonstration under the reperfusion. We would like to represent this contracting pattern in this chart. On the left side, you can see the measuring time. The baseline is at the bottom, the hypoxia is visualized by white, the reoxygenation with gray, and the follow-up period with black color. The contracting activity is visible on this diagram according to this coding. Excluding the time-match control that was not exposed to hypoxia, a similar pattern was observed. The improvement occurred mainly at the reoxygenation period in the contractility, so we focused on this time. In this chart, you can see the registered frequency under the reoxygenation period. 10 to minus 8 mole SNAP and 10 to minus 8 mole BMP provided significant protective effect, and they were able to recover the contractility. Regarding to the rate force product, the improvement was the highest at the presence of SNAP at 10 to minus 7 and 10 to minus 5 mole concentration. However, the BMP was the most effective at the same 10 to minus 8 mole concentration as at the frequency. After the reoxygenation, the level of the cardiac troponin I biomarker was measured. Comparing to the time-matched control, hypoxia caused tendency to rise in the troponin I values. Although no significant difference was observed, there are increased values in some of the samples. In conclusion, we can say that we successfully established a well-reproducible hypoxia reoxygenation drug screening system in engineered heart tissue, and we verified it with well-known cardioprotective materials, SNAP and BMP. The clinical importance of this in vitro method is quite valuable because we can gather information in connection with functional parameters of the cardioprotective materials. Finally, we would like to express our gratitude to our consultants, Dr. Anikor Görbe, Assistant Professor, János PhD student, and the coordinating professor, Dr. Peter Fernandi and Dr. Tamás Esenhagen. Thank you for your attention.